This is Jessica. She's 8 years old and she has cystic fibrosis, which means that she suffers from severe breathing problems and will probably not live past the age of 20. All this because of a defect in just one protein. But what if I told you that we could tell Jessica's cells to produce a healthy copy of this particular protein? You see, our cells are like huge factories, and like any factory, they require a set of instructions telling them to produce a particular product or a protein. These instructions come from the control room in the cell, or the nucleus, and we call these instructions messenger RNA. Thanks to the advancement of technology, we can now synthesize millions of copies of this messenger RNA. And so we have the message that we need to send to Jessica's cells, but the question is, how do we get it there? Messenger RNA is very fragile. If we were to just inject it into the bloodstream, it would just get degraded. Furthermore, it is very difficult for these messenger RNA molecules to get into the inside of cells, which is where we want it to go. And so this is where our research comes in. We at the Macmillan Laboratory have formulated what we call nanoparticles, tiny balls of fat that are able to package genetic material and get them efficiently into cells. The analogy I like to use is like sending a letter in a bottle. If we were to just throw this letter out into the ocean, it would just get wet, damaged, and degraded. However, if we were to put this letter into a bottle, then it will be protected, and the message will be more likely to reach where we want it to go. And so, these bottles represent our nanoparticles, which have already been trialed and have shown remarkable success at switching off the production of certain proteins. My project, however, focuses on getting cells to produce a particular protein and to determine the efficiency of using these nanoparticles at delivering messenger RNA into different cells growing on a petri dish or a plastic plate. Once we've established how well they work, the next step is to trial these nanoparticles in a living mouse model. And in particular, we are looking at the delivery of these particles to different organs over a period of time and at different doses as well. If we are successful, then the therapeutic applications of this delivery system are endless, and ultimately, it will take us one step closer to the treatment of genetically based diseases, such as hemophilia, cystic fibrosis, and cancer. Thank you.